Okay, so good morning and good afternoon, everyone. It's my great pleasure to open this joint webinar organized by the American Ceramic Society and by the European Ceramic Society. This event brings together speakers from bioceramic industries in both the United States and Europe, highlighting the fruitful cooperation between the two ceramic communities. I'm Paola Palmero from Politecnico of Turin in Italy. I'm the chair of the Bioceramic Network of the European Ceramic Society, and I have the pleasure of moderating this webinar together with my colleague, Dr. Hrishikesh Kamat, who is the chair of the Bioceramic Division of the American Ceramic Society. First, I would like to warmly welcome and acknowledge our four distinguished speakers, Dr. Emiliani, the product manager at Finceramica in Italy, Dr. Pradhan, senior scientist at Glidewell Laboratories, California, USA, Dr. Oberbach, managing director of Mathys Orthopaedie in Germany, and Dr. Fu, principal scientist at Corning Incorporated, New York State. This presentation will cover a broad range and exciting range of topics related to bioceramics and bioglasses for industrial and clinical application, including glasses for healthcare and ceramics for dentistry, for orthopedics, and for regenerative surgery. Throughout the session, we will explore the path from research to industrialization, discuss recent innovation and clinical use, and reflect on how these fields have evolved from the past solution to future perspectives. A key topic of today's event is also the dialogue between the industry and the academia, which is essential for progress in the bioceramic field. Before we begin, I would like to take a moment to acknowledge a few colleagues who made this event possible. So Professor Annabelle Brun from KU Leuven in Belgium, and Professor Jérôme Chevalier from INSA of Lyon in France, who actively contributed to building the program. And Dr. Vicki Evans from the American Ceramic Society, who is providing technical support during the webinar. And of course, the leaderships and the staff members of both the American Ceramic Society and European Ceramic Society, as well as all the members of the Bioceramic Division of both the ceramic societies. Just before starting, a few practical notes. Each presentation will last around 20 minutes, so you are all invited to ask your question using the chat function. After each talk, we will take a few minutes to read and discuss some of your questions. Anyway, all questions will be also collected, and our speakers will kindly provide written answer afterwards, which will be shared with the participants. Once again, thanks you all for joining us today. I'm confident that this joint event will inspire new ideas and strengthen collaboration across our international bioceramic communities. And now I'm pleased to give the floor to my colleague Rishi, who will introduce our first speaker and start the session.
Hey, thanks for the warm introduction, Paola. So good morning and good afternoon to all of you once again. I would like to now invite our first speaker, Dr. Nicolas Emiliani. So Dr. Emiliani is the product manager of Custom Bone, the line of custom-made cranial prostheses developed by Finceramica Fienza SPA. He holds a master's degree in biomedical engineering from the University of Bologna and a master's in project management from the 24-hour business school in Milan. Before joining Finceramica, he worked as a research fellow at the University of Bologna within the bioengineering laboratory of Politecnico Sant'Orsola Hospital, focusing on 3D printing of materials for high-fidelity surgical simulators and on medical imaging processing. He has developed advanced technologies for preoperative planning and also tools for use of virtual reality for surgical training and augmented reality systems for intraoperative navigation. With that said, it's my pleasure and to welcome him and for all of us to learn from him about innovative solutions for regenerative surgery in Finceramica. All to you, Dr. Nicolas.
Thank you so much. So good afternoon and good morning to all. Thank you again for the invitation and thank you so much also for the presentation. I can share my screen. So are you able to see my screen? And can you hear me well, right?
Yes.
Okay, good. So thank you again to all of you. I'm Nicolas Emiliani. I'm a biomedical engineer and product manager at Finceramica in Faenza, so in Italy. And in this presentation, I will show an overview of our company, our business core, and which are our future direction where we're going on. So just regarding the origin, Finceramica was founded in 1992 as a spin-off from the Institute of Science, Technology and Sustainability Development of Ceramic Materials. So this institute is an institute of excellence for the ceramic research and it's part of the National Research Council. It's based in Faenza. Faenza is located in the northern part of Italy between Bologna and Ravenna. And it's a small town. We are talking about more or less 60,000 inhabitants. And you can ask you're wondering why Faenza. Faenza has a very long history and a very good tradition regarding the ceramic. We are very well known worldwide for our artisans that works and produces the famous Maiolica Faentina. So this is the right town to do this. But after the foundation in 1992, a big step in the medical field was in 2005. So in 2005, the Tampieri family, who owned the Tampieri Financial Group, acquired Finceramica. So even if they are not involved in the medical device field because they're mainly involved in a complete different of seed-based oil, and they're also active in the renewable energy. They wanted to acquire Finceramica, and from that year we started playing a very important role in the medical device field. So the Tampieri is a multidisciplinary group, and they are a very huge company. The revenues are around $400 million per year. And on the left of the slide, you can see all the company that they own and they manage. Now we can go. We can our philosophy of Finceramica. I reported here a sentence from Anna Tampieri, who is one of the founders of Finceramica. So we can say that the starting point is the clinical need. Then we need an innovative and or an original idea, which is effective and has a good cost-benefit ratio. And then we can move to the industrial research to bring the therapy to the market. But most important, the second sentence enclose our philosophy. So we want to change the perspective from the concept of repair and replace to that of regenerate the bone. So regenerate the bone is our main philosophy that all our products have. So our mission is to design, develop, and distribute state-of-the-art therapies for bone and joint generation and joint regeneration, providing patient-specific solution worldwide. We want to be a leader in biomimetic devices and in general regenerative medicine. And we have values that are shared also with the Tampieri family, like the quality and the scientific excellence, the passion for science and the expertise, the compliance and the transparency, and last but not least, the teamwork and the partnership. So as I told you, our philosophy is to regenerate the bone. And how do we approach to tissue regeneration? all our products that we have in our portfolio are based on three key properties, which is the biomimetism, the intelligence of materials, and the nanostructure. So all these properties help the body to regenerate the damaged tissues. And going more in deep, so the biomimetism is the more similar a synthetic material is to the native tissue, the higher the chances of to have a successful regeneration. All our materials are nanostructures, so are characterized by specific nanostructures that help repair the damaged tissue by acting at the molecular level. But they're also intelligent materials because they are designed with a specific geometry and a microarchitecture that allows them to act as a scaffold to guide the tissue regeneration. So I'm very happy to present our products, our solution for bone regeneration. We are mainly involved in three areas, which are the orthopedics and the spine surgery and the neurosurgery. So starting from orthopedic and spine, we have family of products for the bones, which are bone substitutes. We have three different line of products. So NG-POR. NG-POR is a preformed bone substitute made by hydroxyapatite. We have the differentiation between spine and ortho. And we provide different shapes of NG-POR. So starting from the chips, as you can see in the slide, but also cylinder or rectangular shapes, or for the shoulder, it's a trapezoidal shape. So different shape, but also different sizes. Then we have SynthLife, also differentiated by spine and ortho, which is a moldable bone substitute, so a putty made by hydroxyapatite and enriched with magnesium. And Regenos, which is a flexible bone substitute. Also, Regenos is differentiated between spine and ortho. And also for SynthLife and Regenos, we have different sizes and different sizes. Then we have MyRegen. MyRegen is our device for osteocartilaginous joint repair. As you can see, we have three different line of products. So the Prime, Slim, and Chondro. Here is represented the Prime, so it's a scaffold with 3 layers, because the Prime is the one indicated for severe osteochondral lesion. So we have 3 layers because we want to mimic the different layers that we have in our body. It's indicated for knee, ankle, and all the other joints. Especially, it's especially used in knee cartilage repair. So the 3 layers, the upper part is the one that has to mimic the cartilage. So it's made by equine collagen, so coming from horses, while the second and third layer are the ones that have to replicate the bony parts, so are composed by collagen, hydroxyapatite, and magnesium with different balance. This is the Prime, while the Slim and the Chondro, the Slim is indicated for less severe osteochondral lesion. So it has 2 layers, the first one and the middle one, while the Chondro Plus only for chondral lesion. So there's only the collagen layer. Last but not least, we have also Custom Bone. Custom Bone is our patient-specific cranioplasty device. Is made by hydroxyapatite. Custom Bone is the name of the product for all the world apart from USA. In USA, the name of the product is Customized Bone, but the product is the same. For all of these products, we have a lot of data because we are on the market since 20 years. So we have a lot of data and a lot of published papers for all of these products. Just an example, I'm more focused on Custom Bone, and we have almost 80 published papers regarding Custom Bone. So we perfectly know the behavior of our product regarding all the aspects of Custom Bone and evidences of any doubts that the surgeon can have in using our product. So all of these material are covered by patent, like the custom bone has a patent which covers us in more than 20 countries. For the bioceramics and the moldable and the putty, we have 4 patent families, while for the nanomedicine device, so the like myoresin, which involves the collagen and in combination with hydroxyapatite, we have 5 patent families. So here are just where we are covered by our patents. So let's say worldwide. What we are focused on, so our products have all of them. Some of them are we still are in transition from MDD to MDR, but the other one has all the MDR. Custom bone has also the FDA approval. Myoresin doesn't have the FDA approval, and the company has decided to invest on this. So we are organizing the clinical study for the FDA approval. So we are very busy in organizing all the study because it's a very long process, as you will see also in the next slide. So this one, the other topic that we are focused on is the antibiotability of Regenos. So we are waiting for the MDR approval for the antibiotic Regenos. And the other topic that we are focused on is the market expansion. So our regulatory affairs department is very busy with the registration of our products in new countries because we want to expand our market as far as possible. So we are very busy also on this side. As I told you, our products has a lot of published paper, and all of this could be possible and is still possible because of our continuous collaboration between our company and the institute where we spun off. So we're still collaborating, and in several nanomedicine and tissue engineering R&D projects, and these are all of some of the international and national projects that the company participated in. And so over the year, the institute has been involved in a lot of national and European research projects, and has produced a substantial number of publications. So we have had the opportunity to collaborate in many of these projects, and which have also supported the development of our new technologies. And this is very important because I really believe, I come from the academic side, so I really believe that the strength of collaboration with the academic and the industrial companies lies in increasing the company's know-how thanks to the open-mindedness that we can find in the academic area, but also the knowledge of technologies and experimentation. So I think it's a very important and fundamental point to the collaboration between the companies and the academics. So this is just an overview of how multidisciplinary is the development of medical technologies. So it's an iterative process that incorporates the clinical, the preclinical, but also the manufacturer, the production, the designer. So it's very important to have all these aspects in mind because we want to have the high quality standards. So to ensure this, we operate under strict regulatory frameworks. So all our devices are manufactured in compliance with the GMP, so the Good Manufacturing Practice Guidelines, ensuring the complete traceability from raw material to post-market surveillance. And I want to highlight the post-market surveillance because the life of a medical device doesn't end when I sell the product to the customer, but the post-market monitoring is crucial to confirm that the device is continues to be safe and effective over time. And why I highlighted this, because with the advent of the MDR, everything changed. There has been a sort of revolution in the medical device field. So the adaptation to the MDR requires a significant investment, but not only in terms of money, but also in terms of time and internal resources. So it demands a more clinical data, a more robust technical documentation, and continuous monitoring of product performance on the market. So this effort allows us to position ourselves at a very high level. And just to give you an example, some competitors that we had in the past are not any longer on the market because they did not have enough clinical data and also probably also money. We are lucky, but also we always operated in following the guidelines. So all our products are MDR approved or in transition between MDD and all the guidelines. Just an example regarding also the big amount of money that we need to develop for the development of medical devices and also biologics in US. This is an example, but we need to invest a lot of money to have a result in a long time period. So for the medical device, we're talking about 8 years, biologics still longer with a huge amount of money. So for example, our myoregion, we are quite advanced phase, but we are not going to launch if the study will be okay or the outcome will be reached, we're not going to launch on the market myoregion in US before 2030. So it's a very long period and a long story. And I wanted to highlight that with the MDR, the regulatory aspects are becoming very important, much more important. So because you always have to provide data and to prove from the intended use of the product, the composition, you need to verify the product compliance, then you have to evaluate the performance before in an animal study and then in a clinical study. And then you have the approval for commercialization, and it's not finished here because you need to have all the data from the post-marketing surveillance and the data acquisition and so on. So, conclusion, the take-home message are that each product that you have must be you need to show the clinical safety and efficacy of each product. And if the product does not work better than the standard of care, so you need to review also the commercialization strategy, and you're in trouble in that case. We are always open to collaboration. What we look for, it's for sure the people who have an experience in multidisciplinary field, so regulatory, clinical, engineering, and so on. Obviously, the knowledge of bioceramics and biomaterials and other topics that are quite important, but not fundamental. But I wanted to say that we are open and we have experience also with master thesis. We have experience in collaboration with the PhD students. So I think that every opportunity is important both for the company and for the individual to grow and to have new ideas to bring on at the light. So thank you very much. I placed here the link to the website of the company and also my email. So if you want to contact me for a possible collaboration, I'm open to discuss and also with my colleagues. So thank you very much.
Thank you, Nicolas, for that insightful presentation. We really appreciate sharing your expertise and talk in today's meeting. So we will now quickly move on to the questions from the audience. So I have one question here. So it's concerning 3D printed devices. Can you say a few words about the necessary compliance to MDR regulations compared to standard bone substitutes, and how many 3D printed implants have already been implanted to patients?
So they're not 3D printed because first they're not 3D printed because hydroxyapatite, I think that in Europe there are some companies that are trying to 3D print hydroxyapatite, but it's not so easy. So first, this one, they're not 3D printed, but we have other traditional technique, let's say, and we have a patent also on hydroxyapatite, so I cannot say too much. But
Okay.
regarding the MDR regulation, I know that regarding the bone substitutes, some of them are already MDR approved. And I think we are waiting for only for the Regenos, but we are on the market since 20 years, so we have, as I told you, we have a lot of data, and we should not have any problems. Every time we talk with the notified body, we are quite sure that there are no problems. And I don't know how many implants were already implanted because I'm more focused on custom bone. I can say that custom bone, we sold more than 10,000 implants from the beginning of the 21st century, so the 2002, more or less. We sold more than 10,000 implants. And okay, I'm looking at the chat, so okay, the 3D printed, the personalized implants. No, also custom bone is not 3D printed, but it's fabricated with the traditional technique.
Okay. Thank you so much. So if we have any further questions, maybe we'll collect them and send them to you. But thank you, Nicolas, once again for the talk. So now I'll hand it over to Paola, who will introduce our next speaker for the seminar. Thank you.
Yes, thanks so much, Rishi. So it is a pleasure to introduce the second speaker today, so Dr. Dimple Pradhan. So Dimple is a senior scientist at Glidewell Laboratories, leading research and development for next generation delta material. Since joining Glidewell in 2016, she has contributed to the development and launch of several products, including Bruxir Zirconia and Bruxir Aesthetic. Several of her innovation has been filed as patent application. She brings strong project management expertise, especially in design control for medical device with a focus on FDA clearance. DIMS leads a team of scientists, engineers, and actively mentors students through the Make a Difference Opportunity Program of UC Irvine, offering hands-on industry experience. Dimple earned her Master of Science and PhD Material Science and Engineering from Alfred University, where she also worked as an adjunct professor and received the 2012 Dr. Richard Martin Outstanding Teaching Assistant Award. So it's a pleasure now to learn more about ceramics in dentistry, present and future. So I leave the floor to Dr. Pradhan. To Dr. Pradhan, please.
Thank you so much, Paola. I hope I'm audible. I'm going to share my slides now.
Yes, okay.
Please let me know if the slides are visible.
Yes.
Okay. Thank you so much, and everyone, and good morning and good afternoon to all the audience and the committees. My name is Dimple Pradhan, and today I'm going to be talking about ceramics in dentistry, present and future. The outline of my talk will be as follows. First, I'll be introducing myself and give a brief history about me. Second is I'll be venturing into Glidewell as a company and Glidewell product. Then we will peek into restorative dental market, followed by a deep dive into our flagship product line, which is Bruxer family. I'll also briefly touch upon the chairside dentistry, which is our Glidewell.io product line. Then I'll pivot the presentation towards transitioning from academic to industry, for which I've broken down for simplicity into the number of years I've worked in Glidewell. And then finally, I'll provide an insight to what Glidewell can offer if you guys plan to join Glidewell. So moving on to a little bit about myself. So I was born and brought up in Qiang, Sikkim, which is a small state in India. And this is me with my parents during my childhood. So coming from that place, now currently where I reside is in Southern California. And here I am residing with my better half, Hrishikesh, Kamat. And a little bit additional detail of what my life is. I love to hike and I love momos, which is Sikkimese dumplings. In terms of career footprint, I graduated with bachelor's in ceramic engineering from NIT Rourkela. There I graduated with silver medal from, and this I received from the hands of Dr. Abdul Kalam, the president at that time. And during my bachelor's, I got an opportunity to work at different internships. So one such was CGCRI Kolkata under Dr. Amarnath Sen, where I worked on gas sensors. Then I also got an opportunity to work with Dr. Parag Bhargava at IIT Mumbai as an intern on co-precipitation of YSZ ceramics. After that, I moved to Alfred University for my master's. And my master thesis was on structure and biocompatibility analysis on sol-gel prepared niobium and titanium oxide. There are some articles that are published in those lines. I also pursued my PhD in the same university, and we had some articles published in those lines as well. After my PhD, I moved to Glidewell, which is where I'm currently at. I joined Glidewell in 2016 as a scientist, and I moved the ladder, and currently I'm a senior scientist over here. So I work actively in innovations, dental material research. I'm also involved in product development, and I also dabbled into project management as one of the opportunities in Glidewell. So with that being said, these are a few product lines that I have contributed in, and there are a few patent publications that I have been a part of. So moving on to Glidewell as a company, it was envisioned by our founder, Jim Glidewell, in 1970, and the mission of Glidewell is to increase patient access to premium dental services through improved quality, convenience, and cost effectiveness. This is our Glidewell campus, and as you can see, we have the whole R&D division along with the manufacturing facility. We also have the clinical research operatory, where a lot of our materials are tested first in hand internally. And this is our Glidewell R&D family. And if you guys are trying to locate me, this is where I am. So in terms of products that Glidewell offers, we offer a diverse portfolio of products, starting from crowns and bridges to Glidewell in-office solution, which consists of not just the milling materials, but also the fast mill is made in-house. It is a mill that is the size of a microwave that can mill those materials and seat the patient with a crown. Then our latest addition is the Orb, which is a smart wearable device which tracks heart rate, body movement, and impact and is used for sports person. So now moving on to US dental restorative market. As you can see over here, it is a 10 billion market, and crowns and bridges offers around 6.7 billion in revenue, and it's about 65% of the market share. The main players over here are zirconia, glass ceramic, and PFM. And zirconia, which is our flagship product, which is Bruxer, is a big game changer in this landscape. So if coming to Glidewell product specifically for Bruxer, Glidewell launched Bruxer Full Strength in 2008. It is a greater than 1000 MPa product, and it was a game changer, and precisely why, we'll get into that. So before Bruxer, PFM, which is a porcelain fused over metal, was the choice of material for dentists, but it had its own challenges. Dental porcelain were prone to chipping, and the crown preparation was also very skill dependent. The moment we introduced Bruxer in 2008, we can see a huge change in the market landscape. We can see that by 2020 or 2022, the PFM was declining and Bruxer became the choice of material for the dentist, especially for the posterior regions. Then in 2017, Bruxer Aesthetic was launched, which is about 870 MPa product line. And there we can see that there was a historic shift in the market materials preference for anterior. And why we are saying that is already published in the article in American Dental Association. So what it says is up to 2016, only 5% of the dentists would choose monolith zirconia for anterior regions, mainly because of the aesthetic issue. And by 2021, up to 61% of the dentists started using monolith zirconia for anterior regions. So we are not stopping there. The next launch was Bruxer Radiant, done last year. So we are now gearing up for our next milestone for the remaining 39% of the dentists. And Bruxer Radiant, what it offers is the strength of zirconia designed to match the beauty of glass ceramic. The translucent contact regions are just like the natural tooth. If you look at the natural tooth, it's not a homogeneous body. It has a very translucent incisor and a polychromatic body, and that's exactly what Radiant offers. And the dentists are already raving about this product. Basically, as you can see from Dr. Belderis, they're calling Bruxer Radiant a no-brainer. You're comparing it to the leading glass ceramic and saying that Radiant is 65% strong and 5% more translucent. So how are we getting this, right? Basically, we can see there are two major properties into considerations over here. One is the flexural strength, and second is the optical property, that is the translucency. And if you look at the regular YAZ ceramic in the market, you can see that it starts with the 3Y, which is the most, having the highest strength as possible, around 1200 MPa. And then as you increase the yttria level, you see the difference that it slowly decreases the strength, but improves in translucency. Why does that happen? It's a very well-known understanding in the material science industry, material science field, that as you increase the yttria level, you're going from a more tetragonal phase to a more cubic phase, right? So as you increase the yttria, it's going to undergo a phase transformation. So what is Glidewell doing different here? So as you can see from the Bruxer Full Strength, which is a 1200 MPa product, we are able to have Bruxer Aesthetic as well as Bruxer Radiant. And what it is able to do is we are able to interchange the phases in our system and not have the compromise on the strength or decrease in the strength as much, but increase the aesthetic, because that is what is required for the performance of our product line. How we are able to do that? Well, we focus on various things. First is we focus on the formation of the green body, right? Where we are very much aware of what materials, raw materials to select, because the particle size that you're going to select, the surface area that of your raw materials is going to govern your final property as well as the formation of your green body. Also, the processing method that you're going to do, how you're going to pack your green body better so that you have a better packing density, all those factors are taken into consideration when we form the green body. We also look into finer details of sintering, such as grain size optimization, removal of porosity, and phase optimization that I touched upon earlier, so that the final sintered body is going to give you something that is patentable and something that is going to change the landscape of dentistry. So next is, I want to touch upon a little bit on lab versus chairside. It might be a new term for people, but let me ask you guys a question, or I can answer it for myself, that if you guys have ever gone for a root canal treatment, I have experienced a root canal treatment. I know for sure that that doesn't take place in one day. You have to go for multiple visits to the dentist office, which costs time, money, energy, right? So why does that happen? I used to wonder when I was a kid as well. Well, that is because when you go to a dentist office, they examine, they do their procedure, they scan it, and they send your impression either digitally or physically to a lab, and which might be very far away. So basically the lab is where the crown is actually being prepared, and they are literally shipped like a FedEx ships like, you know, your packages home, or Amazon ships your packages home. Your crown is shipped to the dentist office per se. So that takes multiple days of visit. So what Glidewell was able to do was actually forecast something like this called chairside dentistry, where we offer them a solution where dentists can mill a crown right at their office and patient can go home with a crown under an hour. And something like this actually started getting more momentum during COVID, because dentists we were able to empower the dentist, and they were able to be less dependent on the labs in that case. So how is Glidewell able to do that? What we provide to the dentist are sintered zirconia milling units. And we have a fast mill, which is a size of a microwave. And you also have a milling burr, and they're able to mill this into the shape of a crown. And now that the trademark stands for no oven weight. So these are already sintered, so they don't have to have any furnace or heating time, and it is seated in the patient's mouth. So there are various product lines that we offer in this category. We also offer Bruxanol bridge blocks, which is not just single unit, but multi-unit that can be placed in the patient's mouth as well. So as a material engineer, we have to focus on what are the material considerations when we think about chair side. Unlike the lab side, where we are thinking about the green body and as well as the sintering, of course, those things are definitely there. But since you are milling it in a very aggressive environment, you have to also look into hardness, fracture toughness being an important factor in this case. Because if you look at the margins, you have to make sure the margins doesn't have any of these rigid lines, because those are areas where the bacteria can grow in. So you have to make sure there's no chipping at the margin areas. We also have to think about sintering a bigger mass. Unlike a crown, which is like a 1 gram, these masses could range anywhere from 30 grams or even 70 grams, right? So these are bigger block. So you want to make sure that the center of the block is centered and does not have this kind of porous microstructure. You also do not want the shades to have this kind of inhomogeneous zones. And what is basically desired is a homogeneous microstructure as well as a color uniformity in this case. So sintering definitely is a different ballgame when it comes to this kind of materials. The next thing is also engineering considerations, because if you think about it, the block design, the selection of the milling tool, establishing of the milling forces, and the interaction of materials with the milling tool all needs to be in place to marry the material with the milling machine. So hence, Glidewell is the only company which is able to do that, because we have a strong R&D team as well as a strong engineering team who is able to make this possible. Now coming to the slides where I'm going to pivot a little bit more into academic to industry. And as you can see, based on my career path, I've worked more in academic up until my master's and PhD, and I transitioned to Glidewell, and this was my first industrial experience. So I wanted to touch upon, which might help some of the new graduates or people who are looking into industry as an experience, what would be the insight into those lives. So if you think about in academic or in industry, you set out for certain goals in your project. And when you achieve that, you have a eureka moment, right? And you are like, okay, the goals are achieved. So in an academic setup, normally when you achieve those goals, maybe you can have more papers published, you go for a thesis defense, you have conferences where you showcase your results. In industrial setup, what I understood is that is the first step to do, because after that you have to also establish process repeatability, which is you should be able to repeat your results. But the most important thing was reproducibility, they can reproduce my results, because people who are not skilled in the art, let's say you have an operator or a technician, can also follow your process and come to similar conclusions. So the R&R game has to be really strong over here. And what do we need for the R&R game to be strong? First of all, you have to make sure that you remove ambiguity in the process. You develop clear procedures and SOPs and protocols so that people can follow the process and come to the conclusions. You have to also look into calibration of the equipments. You have to also make sure that the operators are trained in the process. And the primary most important thing is you have to make sure you have an acceptable variation, establish an acceptable variation in your process. Because when it comes to reproducibility, it is not about getting the exact values. It is about making sure the data is within the acceptable range and meets the performance criteria. Second thing is, why is establishing acceptable variation important, right? Imagine a process with such a small working window, with such tight tolerances. It would be a manufacturing nightmare, right? How will anyone even replicate something like this? And what it will also do is it is going to increase the cost and time, because it's going to have more rejects and reworks because your acceptance window is so small. It's going to be a biggest challenge when it comes to scaling it up, right? So another thing, what is also important as an industrial scientist is you have to establish a good working window so that you can meet the manufacturing requirements or manufacturability requirement per se. So next thing is when you're scaling up, right? So let's say your R&R game is strong, you hit the bullseye multiple times, and you also establish reproducibility, and your operators are trained and they are able to follow the process and meet the performance criteria. The next thing is, can you scale this up? And that's a big question in the industry, because you work at R&D at a prototype level, and when it comes to production, you need to be able to scale it up. So what are the things that needs to be kept in mind when we do that? First of all is, of course, your batch sizes are going to change, right? So you start with maybe 100 grams or 2 kilo batch at the most at R&D level. When you're scaling it up, it changes to 2 kilos or even a few tons. Your equipment sizes are going to change. Your R&D furnace might be putting in like 10 samples at a time. And when you're going for manufacturing furnaces, it would be like 5,000 samples at a time. Your personnel might also change, and actually will change, because your R&D personnel are people with whom you have started the research per se, right? And then you have your manufacturing operators who are new, who need to be trained in the whole process again. So in all this, it is very important as a SME to identify what are the critical process parameters, and also establish correlations between R&D and manufacturing, whether it comes to equipment or whether it comes to your process controls. And most important thing is also to understand manufacturing constraint. What I mean by that is, let's say at the R&D level, you developed a process which might be very, very complicated. It might be 15, 16 steps, but is that scale up friendly, right? So there, as a SME, you need to start proactively thinking about how you are going to start cross-functional collaborations. Think about, is there something that can be automated? Then you bring in engineering. Is there something that can be done differently? So you need to have that open mind and open mindset to start that initiated discussions with the whole team, and not be my way is the highway kind of person. Next is, I want to go through the different stages of product development. So whenever something has a prototype development and you did a manufacturing feasibility, you undergo something called as a phase gate or review board of approvals. So during these meetings, the technical discussions are already out of the door. It is already understood that you, being the SME, you have your PhD, you have all this research in place, you know what you're doing, right? So you don't need to get into the technicalities over here. What the business is actually interested is, what is the timeline? How fast can we enter the market? What is the budget? So that the budget ties to the return of investment. What are the COGS? What are the resources you need? Because you're not going to be a one-man army right now. You're going to actually work with manufacturing, marketing, regulatory, like technicalities with engineering. So you need resources from everyone. So that is something that you have to make sure that the review board approves. And that is the first phase of product development. The second phase is, and because we are a medical device company, the FDA provides design control guidelines. So what that is it's a PDF. Anyone can actually take a look at it. It provides a guidance as to how to like develop a product in the for the medical device. So you start with the planning, which is identifying the scope of the project, and you have your users' need met, right? You identify what is the reason you want to make this product. Then it undergoes a design input where when you set certain product specifications, and you have your design process followed by design output, where you identify and establish what your design output is going to be. You also have a design verification where you demonstrate the product meets the input requirements, and you have your validation where you see that the medical device meets the user requirement. So because we are in the dental industry, and we undergo the same design control process, we have an ISO that we follow, which is the ISO 6872. That's our Bible. It kind of sets a series of experiments or testing that needs to be in place to make sure your device is in compliance to the FDA guidelines. So there is radioactivity, flexural strength, linear thermal expansion coefficient, glass transition temperature, chemical solubility. This, in addition to many other things, like for example, because you're going to put the device in a human mouth, you have to make sure it is biocompatible. So there could be other biocompatibility tests like cytotoxicity, sensitization, and all those in place to make sure that your device is in compliance. Next is all the documentation of the input, output, verification, and validation stage undergoes something called as a compilation stage known as design history file, where all of these are documented in proper form, and it undergoes a review with not just R&D, but also with RA, which is the regulatory body, as well as QA body and manufacturing, and whoever people are involved with that process, right? So these documents have to be maintained, and this will be facing regular audits. So there should be a proper traceability of these documents. The next thing is anything that is you're going to enter the market most probably will require a 510(k) clearance. So what that is everything that is documented in this case, the records are submitted to FDA in a proper format that is designed by FDA to demonstrate that the new device is substantially equivalent in safety and effectiveness to the legally marketed device. So where we come in and what is the role of like as a SME or a researcher is that because you are the person who initiated the project, you know the top, middle, and bottom of the project. So when the FDA sends out any kind of queries or any interactive reviews are in place, the SME is the one who's going to answer every queries that is pertaining to the history of the product, regarding safety, regarding effectiveness, regarding any testing. So once we give the 510(k) clearance, that is a eureka moment for us, because then we can now market the device successfully. So things don't stop over there. Whenever you have anything that is cleared, you also make sure that you have a controlled beta testing. And I think Nicolas in the previous slide also talked in his presentation also talked about post-market surveillance, right? So that's exactly what we also do. We have to make sure that mostly whenever you are launching a product, you test it with a control group, which are real user and real work environment, and before it is released to every other customers in the market. So what that control release does is it creates a feedback loop. So you've submitted a tester, we understand what the challenges are, what is good, what is bad. So the company gets the review and sometimes can respond immediately, and sometimes that can generate a second project within R&D stating that we need to make this better because that is the review that we are getting from the customers. So the review can happen both internal and external. At Glidewell, we have our dental lab. We also have operatories where they are the first-hand people who test it internally. And we also have focus group or key opinion leaders who help us for external beta testing as well. So finally, when this is what goes into a dental industry, or I think mostly any industry that you start research and development, you have manufacturing, RAQA, product marketing, and customers and sales. So this is the whole product life cycle that any product goes through in an industry. So I hope with this, I think some of you guys have got an insight as to what it takes to get from a prototype to a product. And moving on to what Glidewell can offer you guys. So Glidewell has a strong R&D division, and we have different departments that caters to glass ceramic, zirconia, like we talked about, polymers for dental applications. We also have a strong engineering team. We make a lot of mills, CT scanners, and a lot of automation is also done in-house. Glidewell also offers a really good internship program where we can, any academic or engineers who want to get into industrial experience can apply their knowledge that is learned in the school for real world application, and they can contribute to all phases of R&D, planning, design of experiments, evaluation. And this internship programs provides an individual a whole round experience. So that's what Glidewell offers. That's all for me. If there's any questions, please let me know.
So thank you so much, Dimple, for this excellent presentation and impressive work in the company. Very appreciated also the deep insight of material science and its impact on the dental product that you showed today. Yes, we have some questions, some minutes for questions. So the first one is still related to 3D printing. So taking into account the two aspects of the presentation, what is your point of view of potential use of additive manufacturing like stereolithography to process dental restoration like crowns and bridges? Can we expect having additive manufactured products with brilliant colors and enough optical properties compared with mill restoration?
Thank you for the question, Paolo. Yes, 3D printing is definitely, I think additive manufacturing is definitely something that the company is also looking into, not mainly for zirconia materials per se, but more like a composite body where you have zirconia or ceramic or glass ceramic as a filler. 3D printing definitely offers advantage because you can remove manufacturing costs. You can get to the finished product much quicker. So we do have some of our dentures that are 3D printed. I think that is already there on the website. So definitely 3D printing in dentistry is definitely the next big thing, I would say.
Okay, thank you so much. We have time for further questions. So which type of a Brookseer has a higher share in the market?
The full strength is the highest share in the market.
Okay.
Yeah.
Okay, that's perfect. So still one of them. Can you comment on the trend of lithium disilicate glass ceramics versus zirconia in the dental industry?
Sure. I think lithium disilicate is definitely something that is also preferred by the dentist. And mainly because it offers that aesthetic and natural tooth-like appearance. And when Brookseer was launched, where zirconia was launched, and where zirconia is promising is mainly because of the textural strength, and it is more brawn than beauty. And what Glidewell is doing, as you can see with the incoming years, is they're actually attaining more and more aesthetic appearance. So the Brookseer Radiant, actually a lot of the dentists cannot even decipher or distinguish between the two. So I think not many people had even seen that zirconia with a strength of about 700 or 800 MPa can look so much beautiful and aesthetic. So that's where I think zirconia is coming up in the market for even anterior or aesthetic regions. Yeah.
Okay, thanks a lot. Again, I guess that we need to move to the next speaker. So thanks a lot again. As I was saying, we will collect all the questions and send answer in a second moment. I have also a question that is not on the public chat, so I will copy and paste it on that. So this is time to move to the next speaker. So Thomas Oberbach, it's my pleasure to introduce him. So Thomas is Managing Director of Mathys Orthopaedie in Mersdorf in Germany, and head of the Ceramic Competence Center. Mathys is an international company that develops, produces and supplies hip, knee, and shoulder implants. So since 2021, Mathys has been part of the US Medical Technology Group ENOVIS. In the early 90s, Thomas completed a degree in material science, specializing into inorganic non-metallic materials at the TU Berg Academy in Freiburg. He then joined the Kamat company as a technologist for ceramic hip joint components. After Mathys acquired the company, he became head of the ceramic production and founded a ceramic materials development department. There, he and his team created high-strength zirconia-based dispersion ceramics, now used in many Mathys implant systems. So it is our pleasure now to learn more about these materials thanks to the talk, 50 years of clinical application of oxide ceramics in joint replacement. So please, Thomas, the room is yours.
Thank you. Thank you, Paola. I will share my screen. Can you see the presentation mode?
Yes, yes, it's
Okay, good. Then I will start. Thank you for the introduction, Paola. Yes, good afternoon, good morning, good evening, ladies and gentlemen in the auditorium. I will speak in my presentation about 50 years of clinical application of oxide ceramics in joint replacement, and I will try to give you an overview about the past, the present, and the future. Before I start with the ceramics, some facts about Mathys, about our company. As Paola said, we are a part of the International Surgical Unit in the reconstructive segment of ENOVIS. ENOVIS is a big US company. And in our company, in Mathys, in ENOVIS, in this International Surgical Unit, we produce implants for arthroplasty. That means we produce hip systems, knee systems, and also implants for shoulder. And our company, Mathys Orthopaedie, is located in Thuringia in Germany. It's the former eastern part of Germany, and we have two locations. In one location, we produce metal implants and also instruments for joint replacement. And in our location in Würzburg, where I'm located, there we produce ceramic implants, and there we have also an applied research group for the development of new materials or new ceramic implants. In our location, we are 110 employees, and we have a production space of 2,500 square meters. We produce in our location in Germany mostly hip heads. Hip heads from two materials, Dimple ceramic, it's an ZTA ceramic, and also hip heads made of ceramis. It's an ACerS ceramic. I will explain it later. These are the main product group, and furthermore, we also use ceramics, in this case alumina ceramics, for the production of shoulder heads, and also for liners for hip and shoulder. We use our ACerS ceramic ceramis. When we look back in the past and we look back on the use of ceramics for joint replacement, then we know, or yes, there was Pierre Bouton. Pierre Bouton was a pioneer in this field. Pierre Bouton was a French surgeon, and he carried out the worldwide first implantation of alumina hip heads and cemented monoblock alumina cup. That means he was the pioneer in using ceramics in joint arthroplasty. And the components came from a French manufacturer, and also in Germany there were some pioneers, the surgeons Chris and Mittelmeier, and they used also alumina ceramics, and in 1974 they implanted metal stems, cementless metal stems, with alumina heads and also alumina monoblock cups. And the interesting thing was that it was a cementless system. You see it here in the screw. The cup is a screw cup, which was cementless, fixed in the human acetabulum. That means the first ceramic, oxide ceramic, which was used in this field was the alumina ceramic. We also worked with this material. We have this material also in our portfolio, and with our alumina ceramic, our trade name is Bionit. We reach a density in the material which is very close to the final density. To the theoretical density, we have a grain size in the material in the range of 2 microns and the biaxial bending strength of a little bit more than 400 megapascals. That means we pass all the requirements from the standard. But with this ceramic, and this is not the case for our ceramic only. This was the case for all the alumina components when they was used in the hip replacement. In some cases, there were a fracture of the ceramic head and in vivo, and I think you can imagine that such a fracture is a worst case for the patient, for the surgeon, who has to revise this, and also for the manufacturer. And our fracture rate for hip heads made of alumina was in the range of 0.02 to 8%. That means 28 hip heads of 100,000 implantations fractured. And the fracture, also the in vivo fracture of alumina hip heads in the market in the late 70s and also in the early 80s last century, lead to the point that also a second material was introduced in the market for hip replacement, the zirconia ceramic. And with the zirconia ceramic, I think you know it also from the dental part. You can reach a much finer grain size. You have a higher biaxial bending strength or bending strength in general, and you reach a higher K1C, a higher fracture toughness. And so this material was used for a long time, for more than 50 years, in the hospitals by the surgeons. And this is an overview from the Australian registry from 2015, 10 years old. But the revision, the zirconia heads showed the highest revision rate in comparison with hip implants with an alumina head or also a mixed ceramic head. And the reason for this higher revision rate, after 7 to 8 years, you see the revision rate increases, was that the zirconia ceramic showed the LTD, the low temperature degradation. And this led to a penetration, to a cup penetration, to a penetration of the head in the cup. To explain it a little, here we had the possibility to investigate an explanted zirconia head from a French supplier, from a French manufacturer, and this head was 17 years in vivo in the patient. And normally, when such a head is zirconia head is produced, and before implantation, the tetragonal and cubic phase content is approximately 95%, and the monoclinic phase content is approximately 5%. And we investigated this head, and we found on the surface that the monoclinic phase content increased after the 17 years up to more than 70%. And the phase transformation from tetragonal to monoclinic is combined with an increase in volume by about 7%. And if this occurs on the polished surface on the hip head, then this leads to such an formation of pittings. That means an increase of the roughness. And if you have an articulation partner, which is a softer material like polyethylene, then this leads to a higher polyethylene wear, to osteolysis, and to a necessary revision of the implant. But the main worst impact of the use of zirconia ceramics for hip replacement was the so-called Procedure recall, which was in 2001 and 2002. There was a French supplier, French manufacturer, and they changed their sintering process from batch furnaces to tunnel furnaces, and they had some batches, two batches, which showed after sintering micropores inside the balls. This wasn't detected after the production, and after 1 year and or 2 years in vivo in clinical use, they had fractured due to phase transformation and also crack propagation from the micro cracks or from the micropores, and so more than 350 head fractures were documented, and this was a cut in the field of using ceramics in joint replacement. So that at the moment, and I think also for the next years, there is no possibility for using high or pure zirconia ceramics, stabilized pure zirconia ceramics in this market, and that's why the manufacturers looked for what ceramic materials can be used, and then in 2003. There we have seen the clinical introduction of so-called dispersion ceramics or mixed ceramics by Ceramtech. Ceramtech is the market leader in this field, and most of the ceramic implants which are implanted worldwide come from Ceramtech. And they introduced in 2003 their Biolux Delta material, which is in ZTA ceramic. That means alumina based and toughened with zirconia. Also in our company, we have thought about new materials, new ceramic materials with a higher mechanical strength in comparison to alumina, with a higher fracture toughness and a higher wear resistance. And we developed also in ZTA ceramic. You see it here, the chemical composition. It contains of 75% alumina, 8% alumina, and 25% YTCP. It's stabilized with yttria, this zirconia content. And on the other side, we also developed an ACerS ceramic, which is zirconia based, also stabilized with yttria, and combined with 20% alumina. And the trade names are Zimerec, and for ACerS, it's Ceramis. And we developed these materials, or these both materials, for the application as hip head or also as liner. And as in the presentation before, it was also said, then it's necessary for the approval as a ceramic component, as an implant component, that the material and also the product fulfill different requirements like biocompatibility. We have checked this for both materials, and they passed all the tests. Also, we had to characterize the material and to check is the material our ZTA according the valid standard, the ISO 6474. And for ACerS, there is no international standard at the moment for this. We had to create an internal material specification, and then that also to check are the mechanical properties like burst strength and wear behavior and so on, are they conform to the requirements. Yeah, and if we look on the material specification, and compare it with the alumina ceramic, our Bionit, then you see concerning the grain size, we reach much lower grain sizes with ACZ and also with ZTA in comparison to our alumina. The bending strength is higher, 2 times higher for ACZ and approximately 3 times higher for ATZ and 2 times higher for ZTA. And also you see with increasing zirconia content, the fracture toughness is increased. And with these facts, we can also reach a better properties with the implants. And one big thing, or one main property is the burst strength of such an hip head to avoid such an in vivo fracture. And you see here a comparison between the fracture strength, the burst strength of an ceramic head. It's for all the tests the same type, the same design of the head, but with other material. And you see with this head type, with Bionit, we reach for this test is also a standardized test according this standard here, ISO 7206. We reach for Bionit for alumina and burst strength of 64 kilonewton. This corresponds approximately to 6.4 tons. We can increase the burst strength of the ball head by using ZTA ceramic. We increase it up to approximately 10 tons, 95 kilonewton, and we can double it in comparison to alumina ceramic, to our Bionit, by using the ATZ ceramic. So we reach a burst strength of 124 kilonewton, which corresponds to approximately 12 tons. That means a lot and strong increase of the strength of the material. It's necessary also to test the wear behavior. We have carried out the tests in a hip joint simulator, and we compared it with our previous investigations of our previous results from the pairing metal against polyethylene. And you see, when we use this as reference, that means in pairing metal head against polyethylene cup or polyethylene liner, then it's possible by changing the material from metal to ceramic, from metal to ceramic, it's possible to decrease the polyethylene wear. It's always the target, the aim, to have a low volumetric wear rate. And then you can also change or can use better polyethylene qualities like cross-linked polyethylene. And with this, you can also decrease the wear rate in comparison to the conventional polyethylene. But also here you can see by using Ceramis or a ceramic head against this liner, you can also decrease the wear rate. And here on the left-hand side, you see the so-called wear results for the so-called hard and hard couplings. That means ceramic head against a ceramic liner. It's called COC, ceramic on ceramic. And you see with this pairing in this test, under standard conditions, the wear rates are much lower than for hard soft, for ceramic or metal on poly. And also with the new ceramics, ZTA and Ceramis for ATZ, we could decrease the wear rate again. But for zirconia ceramics, it's also necessary to investigate the degradation behavior. That's why we have checked and we have tested this degradation behavior by using artificial aging in an autoclave and for over different time from 2 hours up to 400 hours for the ZTA ceramic. And we compared the aging behavior, that means the increase of the monoclinic phase content on the surface, which is determined using XRD and Rietveld or Garvey-Nicholson. Here you can see the Garvey-Nicholson method, and we compared it with published data from France, from Jérôme Chevalier from the late 90s for the YTCP, and you see in the laboratory scale, it's possible, yes, to create a low temperature degradation, but the aging of the ACerS ceramic occurs in the laboratory, but it's 5 to 7 times slower than for the pure YTCP. And for the Simeric, aging is, and this is due to the lower content of zirconia in the material. It's not a big, or it's not a real issue. In the literature, you can find that 1 hour aging corresponds to 1 year, and this is a logarithmic scale here. And if we look on this curve for the YTCP, then you see after 10 hours aging, which corresponds to approximately 10 years in vivo, we have an monoclinic phase content on the surface of about 50 volume %. And as a comparison with ACZ, we have the comparable volumetric phase content concerning Garvey-Nicholson related to 100% zirconia after 40 to 50 hours. That means, and when this corresponds also 1 hour, 1 year, then the monoclinic phase content occurs with 50% after 40 to 50 years, which is not clinically relevant. That means we see aging in the laboratory, but without relevance for the clinical case. Yeah. What about the clinical data, registry data of the implants? You remember the in vivo fracture rate of the alumina heads was in the range of 0.028. And when we look on our vigilance data from our post-market surveillance, then we look back on about 410,000 implanted pieces in total made of Simeric and Ceramis, and we didn't found up to now, and this is a very, very good result for us, we didn't have a fractured head. And so, in our opinion, the problem with in vivo fractures for hip heads is solved with the new ceramic materials which are available on the market. When we look on the registry data here from the German registry, from the annual report from this year, you see that the most heads which were implanted last year in Germany consists of ceramic. That means the percentage of using ceramic in primary hip replacement in Germany was last year more than 90%, and only 5% were from metal. And the mostly used pairing is the pairing like here in this picture, ceramic head against polyethylene or cross-link polyethylene liner, and the ceramic on ceramic pairing. It's more a niche here in Germany. Has only a share of about 6%. That means the target is to substitute the metal implants with ceramics due to the good results. And also when we look in the Australian registry, then we see the revision rates over the years, and the target is to have only a low revision rate. And you see the pairing ceramic on cross-link polyethylene shows after 23 years lowest revision rate of the common use pairing, and also ceramic on ceramic shows a lower revision rate than the other pairings with metal or ceramic head against polyethylene. What we will do in the future. One point, and we are working on this point, is to create a new generation of ACerS ceramic. We call it Ceramis second generation, and the target is to increase the strength and the toughness again for the realization of very thin wall ceramic implants in arthroplasty. Because when we can decrease the wall thickness of the implant, then the surgeon has to the possibility to preserve more bone stock, and so this is one target, and we have some projects at the moment where we investigate this, where we develop this new ceramic, this Ceramis second generation. We want to use also the formation in situ formation of platelets to increase the toughness and the strength again. And potential products are possible for all the different applications, for hip, for knee and shoulder, and I will only focus on the hip. Also here is the target with the ceramic resurfacing components in the hip to substitute the metal on metal pairing. And at the moment there are two very interesting clinical studies with two different systems made of Biolox Delta ceramic from Ceramtech and two companies. And also in our company we have started a feasibility study to use our Ceramis for such an application, but this would be, I think, a very innovative and interesting product for the next years. Let me conclude. Oxide ceramics are successfully used for joint replacement since more than 50 years. I hope I could demonstrate this to you. Ceramic implants show a very good clinical long-term results with low revision rates. The ceramic components, and this we have also seen from the registry and in Germany, and also it's the same in Australia. Ceramic implant components will substitute the metal implants continuously also in the future. And due to the very good mechanical properties of the ceramics from the system zirconia alumina, the potential problem of ceramic fracture is reduced, and I hope it's solved for the future. Before I finish my presentation, also for your information, we offer continuously here in our company student internships, topics for bachelor thesis, for master thesis in the different fields like material sciences, mechanical engineering, and also biomedical engineering. That means if you are interested, contact us, contact me, and then we will check if we find an interesting thesis, an interesting project for you. Thank you for your attention.
Thank you to you, Thomas. Congratulations, excellent talk and impressive work, the one that you showed to us today. I think that we have time maybe just for one question. So do you foresee a rule for some other ceramics in orthopedics as nitrates or other composites?
Yeah, I think this and in the past we had also there were also some activities with silicon nitride ceramics. I think this is a possible way, but we are more focused on in our company on oxide ceramics. This is due to the manufacturing, the disintegrating atmospheres and so on. But I think for us the future will be dispersion ceramics from the system zirconia alumina and alumina zirconia, different compositions, but we think that we will create materials with an increased toughness. But we will in our company focus on this material system.
Thanks a lot. Again,
Okay.
thank you, Thomas. So I
All the pleasure.
give the room to Rishi for introducing the last speaker today.
Thanks, Paola, and also thanks, Thomas, for that interesting and detailed talk. Let me introduce our final speaker, Dr. Qiang Fu. Dr. Fu is a principal research scientist at Corning Incorporated. He earned his bachelor's and master's degree in material science from Tongji University in China and a PhD in ceramics engineering from Missouri University of Science and Technology. He completed his postdoctoral research after that at Lawrence Berkeley National Lab before joining Corning in 2011. He is currently also the editor of the Journal of American Ceramic Society. So his research focuses on bioactive glasses and glass ceramics, chemically strengthened glasses and glass ceramics, and the processing and properties of functional materials. And for his work, he has published over 70 peer-reviewed articles and is an inventor or co-inventor on more than 70 US patents, including innovations for Corning Gorilla Glass and other products. Finally, Dr. Fu has received major recognitions for all of his efforts, including American Ceramic Society Bioceramic Division's Tadashi Kokubo Award in 2024, the Varshnaya Frontiers of Glass Technology Lecture Award in 2022, the Waldemar A. Weil Award from ICG in 2016, and the S. Donald Stuckey Award from Corning in 2016 as well, including other awards. So it's my pleasure to introduce Dr. Fu and to learn from him on glasses for healthcare from research, invention to industrialization. Dr. Fu, the floor is all yours.
Thank you, Hrishikesh. Appreciate your introduction. I'm going to share my screen. Okay, so we'll switch gear a little bit. Instead of ceramics, we'll talk about glass. So in my talk, I'm going to cover glass for healthcare, research, invention, and also industrialization. So before I talk about the technicals, I would like to thank another people who have helped me along my career. Jeff Coney at Corning, Tony at Berkeley Lab, and Dr. Rahman at Missouri SNT. So as you all know, bioglass is really a wonderful material. So this picture shows where you can find bioactive glass inside your body. It has been used for heart tissue regeneration, for wound healing, even for oral care. So for hopefully you will never find any use of this material inside your body. So unfortunately for myself, I already started to use this more than 10 years ago. The first kind of bioglass material I used is a toothpaste that containing bioglass. It is really very helpful to treat my dental sensitivity. Kind of my true kind of complaint is very expensive. It's kind of 2 times more expensive than regular toothpaste. And also it takes about 2 months to see an impact. I believe my second use of bioglass containing kind of product could be a lithium dioxide glass ceramics for dental implants. So we'll see when my first teeth start to break, then hopefully that day won't come too soon. So here's what I'm going to share today. So I'm going to talk about research progress on glass for bone tissue engineering. I want to show you some example how we make a strong and porous glass scaffold for large bone regeneration. And also I want to talk about inventions we made at Corning, give you 2 examples what we have done by designing new composition for wound healing and oral care. And on the last topic, I would like to cover some challenges and also opportunities for industrialization of bioactive glass. I want to show you how challenging it is to mass produce a material like this. So first of all, I will start with glass for bone regeneration. So this SB plot shows how the strengths and versus modulus looks like for natural materials and polymers, ceramics, and also bone. If you look at cortical bone, so the strength is much higher, definitely than trabecular bone. I put the numbers here. So cortical bone is has a strength about 100 to 150 megapascal, and also much higher toughness. So in comparison, so trabecular bone is kind of much lower strength of 2 to 12 MPa. So this macro CT shows the power structure of trabecular bone. It's very interesting. So when I first showed this picture, I was exactly 30 years old. Nowadays, after so many years, I start to definitely feel the decline in my bone health. So on this side, so we really want to make something that could match the strengths, at least the strengths of human bone. So one idea is to learn from this human bone, to learn the structure and then mimic the structure. So that way we can have similar kind of performance when we talk about nutritional delivery, talk about bone ingrowth. I think Nicolas mentioned in his early talk, so he used biomimetic method to produce kind of products out of apatite. So we have used the similar process. In this case, we try to mimic the structure of this trabecular bone. So that way we can produce a porous scaffold. So in this case, we have used 3D printing method to produce this porous, also strong glass scaffold. So to improve the strength even higher, so we try to learn from those biological material. And one good example is nickel. So it's made of this brick and mortar structure, give its very high strength and high toughness. So in this case, so we used a freeze casting method to produce this laminar type structure, so which produce very high strength when you test along this kind of orientation. So that gives us hope to have very high strength and strength material for bone regeneration. So based on these ideas, so we developed 3 types of glass scaffold, used a 1393 bioglass developed by Professor Nina Hooper. So we used her composition, and we were able to produce this trabecular or this oriented scaffold made of freeze casting. All these periodical scaffold made by 3D printing. This shows micro CT images and also shows SEM images. Just to give you some idea, so what type of scaffold we can make out of one simple glass composition. As you can imagine, so the microstructure will definitely have an impact on the strength. So when we change from random to this ordered microstructure, so we have found so the strength of the scaffold increases. So this compressive strength versus porosity shows the range of strengths we can make out of glass scaffold. So as you can see, from when the structure changes from random to ordered structure, so the strengths increase from those comparable to trabecular bone all the way to comparable to cortical bone. So again, so these high strength and glass scaffold are made use a 3D printing technique. I think with all this great kind of microstructure and very high strength, that gives us high hope for use as implants for these load-bearing applications. So we collaborated with our kind of friends in hospital. So we actually use scaffold and load them with cells and signals, and then implant them in the kind of large bone defect model. So in this case, so we created a kind of a segmental bone defect in a rat, and then put a cylinder-like 3D printed glass scaffold within this gap. So this is after 1 day. So you can see the clear gap between this bone defect and also glass scaffold. And after 1 month, we start to see kind of bone grow around the scaffold. And 3 months, so this scaffold start to degrade. and then bone ingrows into the scaffold. And after 9 months, basically the whole scaffold seems to be gone, and you can see kind of bone already integrated very well through the gap. And then for the amplitude control, we have noticed there's kind of almost no bridging of this gap after 9 months. So we looked at additional samples. So we found, yeah, after 9 months, almost all these amplitude defects still remain there. So which means we have established a critical size bone defect model for our study. So I hope by showing you this work, I convince you that this scaffold with high strength really have a high hope for uses in this nodal bone defect, particularly for this segmental bone defect application. So this concludes the first part of my talk. In the second part, so I want to give you 2 examples how we design graft composition for wound healing and for oral care. So as you all know, as some of you know, so Dr. Day from Missouri S&T and Steve Qiang, so they worked on borrowed glass fibers for wound healing. So this picture shows how effective the fiber is. You can even see this soft tissue inside these hollow glass fibers. So what we did here is, so we designed a new composition based off aluminum borate glass. So we did a work using 45F5 and 39.3B3, which is borate version of 39.3 glass. Use this as control and compare with our own composition. So we use a full thickness skin defect model with a diameter of 2 centimeters for this study. So we found, so it looks like our composition shows better performance than either 45F5 or 39.3B3 with the glass Steve and Dimple used a couple years ago. We also did further study, use this histological staining study to confirm the performance of our material. So we show at day 7, so the gap is smaller compared with other 2 glass composition. And day 14, yeah, it's even better. This is kind of one example. So how we use new glass composition for wound healing application. I also want to talk about another application, another new area we have been working on by designing new composition for oral care. As I mentioned earlier, so I have 2 complaints about the bioglass in the toothpaste. It's too expensive and also it's not effective. It's, I would say take a couple months to see an impact. So what we are trying to do is we're trying to come up with glass composition that is more bioactive than the 45F5, which is used this toothpaste application. And also we want to improve the durability of the glass so that we can make the toothpaste formula much more affordable. So in this case, so what we did is, so we switched the system in bioglass from this sodium calcium silicate ternary diagram. So we look into this calcium phosphate silicate phase diagram. So we identified an area around the eutectic in this phase diagram, and we kind of invented a few composition that looks very promising. So when we compare with 45F5 glasses. Just want to show some data we have collected so far. So in one lab study, so we took 45F5 and our glass composition in artificial saliva. So this picture shows how the crystalline phase changes in 45F5 and our composition after 30 days. So what you notice is for 45F5, we only saw some calcite or nitride formation after 1 month. So we even did experiment up to 2 months. We still not seeing any kind of epitide formation. As you all know, kind of epitide formation is a key criteria to evaluate the bioactivity of any material. So for our computation, so what we notice is that even around 7 days, we start to see a small peak in this accuracy fraction around 26 to theta, which shows a formation of hydroxyapatite. So the peak grew higher in intensity after 1 month. So we believe our material shows a really kind of a superior performance compared with 45S5. So it took months for original bioglass to form apatite. So for our completion, it take less than 7 days. So we confirmed this with SEM. So this image shows for 45S5, after 7 days, you only see this sodium carbonate on the surface of the glass. So this sodium carbonate is pretty common for 45S5, so which is because the glass start to kind of corrode on the moisture. So for our glass composition age, what we found is after 4 days, it start to form apatite. Even if we did not see it in accuracy fraction, yeah, the apatite already forms on the surface of glass powder after 4 days. And after 7 days, so you see more crystal, and also you see them to grow much better in morphology. So which means definitely the crystal start to develop with a longer time in artificial saliva. So we did even further work. So we put an additive in artificial saliva. What we found is we actually can form apatite within 1 day. So instead of 7 days without any additive, so within 1 day, you can see all these particles are covered with apatite. So this kind of rough surface on the glass particle shows the formation of apatite. You can see this on a higher magnification. So we believe this kind of ability to form apatite shows how effective this material is. If you think about 45S5, it take over 2 months even to form any apatite. Now in our material, we can form it within 1 day. To really we see a step change in performance in bioactivity. So that is the second example I want to show you. So what we have been doing at Qiang on new glass composition, how we design kind of new compositions for wound healing and for oral care. So really, I want to show you that we really found something useful that could change the healthcare. As you all know, so something useful is not always great within companies. So we really want to look for something that is scalable and also profitable for the company. So we have to make money for the company. So that's the kind of this brings to the third topic of my talk here. So I want to show you what are the challenges and opportunities for mass producing biogas. If we look at the composition for this kind of traditional 45S5 1393 glass composition versus soda. So you will notice soda has much higher silica. I think one key attribute brought by this high silica is its lower liquid temperature and much higher liquid viscosity. So compare with 45S5, which has liquid viscosity around 24 poise, 39.3, 200 poise. So soda definitely has much higher liquid viscosity over kind of 10,000 poise. That's one reason so why soda is so popular, used in window glass or even for packaging. So the glass really have good forming attributes to make into different form factors. One reason behind that is, yes, soda definitely has high silica, so that helps lower the liquid temperature and also brings up the viscosity. So all this help make the soda much easier for mass production. Another big challenging is the fragility of the glass. So if you look at viscosity versus temperature, so the curve of high temperature viscosity of soda is definitely much flatter, and 45S5 and 39.3, you have very sharp, can you see kind of sharp changes in viscosity along with temperature. the fragility index of this bioglass is also much higher. So compared with Soda9 with 45S5 and 39.3, you can see this increasing fragility with the different glass composition. So we use this Mayaga model to fit this viscosity data in order to get a fragility of glasses. I think all these kind of challenges we have been facing when we talk about mass produce of bioglass, how it kind of compares with commercial glass like Soda9. So definitely the scaling up of bioglass is very challenging. But also, I think that's good news. That's great for those of us who work in industry. So which means the company has to rely on us scientists to solve this challenging problem. That's good for the job security for us. So to sum up, so hopefully I will show you that bioglass really demonstrate excellent potential for both soft and hard tissue regeneration. I also give you examples of how we can design new glass composition that can enable a step change in bioactivity that could kind of bring kind of a disruptive product on the market. And also I showed you some examples of how challenging it is to mass produce bioglass in an industrial setting. I think that's all from my talk. I think I still have time for questions.
Yes. So first of all, thank you, Dr. Fu, for enlightening all of us with the different applications of glasses and glass ceramics in the field of biomedical science and engineering. I have a question that I'm going to ask you now. So since you talked about developing the proprietary bioglass composition for toothpaste, so is your team working towards commercializing that bioactive toothpaste? And are you guys looking into going through an FDA process for commercialization? And what hurdles you think you might expect when you are trying to commercialize such a product?
That's a very good question. Yes, we are very interested in commercialize this composition for oral care application. So we are actually working with 2 customers on the market, trying to kind of work with our customer to commercialize it. So for FDA, that's a very good question. So for the toothpaste I talk about, so Sensodyne with noamine, so that toothpaste is not available in the US market. There's kind of very complicated reasons behind that. So FDA definitely one part of the problem. For our product, I think we are working with our customer. So we are kind of trying to see kind of what is the right strategy for this product. Do you want to go through FDA or we want to focus on non-US customer? So that's the topic we are kind of still working closely with our customer.
Okay. So there's another question. So since you have spent a lot of time in academia and then a lot of time in the industry as well, what was the single most learning, if you might want to say, that you learned when you moved to the industry from an academic background?
That's a very good point. Yes. Yes. I think very interesting transition from academia to industry. I think Dimple talks about this, kind of how we care more about publication, new findings in academia, and then in industry, really we care about how to repeat the work and then how to reproduce what we have found. I think another I can add on that. So my kind of key learning is in industry. So we have to make a product to make sure the company is profitable, but we also focus a lot on fundamentals behind the product. So the science behind the product is as important as the skill up and mass production. I think within Qiang, really we put a lot of kind of money and funding into fundamentals between every product. Yeah. So that's a learning I got from kind of transition from academia to industry. So product is important. Fundamental behind the product is as important.
Thank you, Dr. Fu. So one final question. What are some of the opportunities for, say, current engineers, students who are looking into, and researchers who are looking for careers at Corning in the field of biomedical science or material science in general?
Yeah, very good point. So within Corning, I think when we talk about working in industry, so we are looking for people who have demonstrated very high potential through their work, either in PhD or master thesis. So one criteria is the depth and the fundamentals of their research work. So that's kind of one key criteria we are looking at for a new hire. I think the work, the topic, kind of people are working on are not that critical, either glass or ceramics. These topics are not super critical. I think that the depth they have shown is more important.
Okay, so thank you once again, Dr.
Thank
Fu,
you.
and also thank you to all the distinguished speakers today. We truly appreciate your time and effort in preparing these presentations, in delivering these talks to the audience. Before we call it a day, I would like to ask Vicki to share something. So the ACerS, American Ceramic Society, next year in spring. Vicki? Let me, yeah. So I just wanted to let all of you know that 6 divisions of ACerS, including the Bioceramics division and the other divisions, are coming together for the first time next year to host a joint conference that is going to take place in April in Washington. And the abstract submissions for the same is now open, and I think the deadline is 1st of December. So I encourage you and also to kind of submit abstracts, consider submitting abstracts to this conference, and also spread the word with your colleagues and others in the field. So that's one thing. And also I would like to first, lastly, thank all of you for taking time from your busy schedules for attending this event. On behalf of the American Ceramic Society's Bioceramic Division and the Bioceramic Network of ECerS, with me and Paola and Vicki, we again thank all of you for attending this webinar, and have a wonderful day. If one last thing is that we have already collected all the questions that you guys have asked to our audience members. We couldn't unfortunately take all those questions to the speakers in the interest of time, but we will send them out to the speakers and they will kindly respond to them. Also, we have recorded this webinar, so if any one of you wants to revisit this webinar or want your colleagues to visit this webinar, the links for those will be shared on the official YouTube channels of ACerS and ECerS, and all of you shall receive emails once the link is uploaded. So thank you once again. Paola, do you want to add something?
No, just to thanks also by my side all the speakers, and yes, for excellent presentation. Thanks again for your time, and yes, also of course thanks a lot to you, Rishi, and to Vicki for the organization, for sharing this very nice event.
Thank you. Have a nice day.
Thank you to everyone. Bye.
See you.